Okay, so can everyone see my screen? Yeah. Okay, so first of all, thank you very much, Jorge, for this invitation to present to the Argentinian glial community, which I'm very excited. And it's also very nice to see you after all these years. Me and Jorge, we did our PhD together at the same time in different labs. I was in Ernest Arenas lab and Jorge was in Patrick Enfor's lab. Okay, so as Jorge said, my lab is focused on oligodendrocytes and also we are focusing on transcriptional and epigenomic states of oligodendrocytes, both in development but also in multiple sclerosis. So okay. Let me just see. As many of you know, oligodendrocytes are glial cells in the central nervous system, whose one of the main functions is to produce myelin, which is this lipid-rich membrane that insulates the neuronal axons and allows fast transmission of electrical impulses by the neural circuitry. Now, this is a cross-section of an axon with the myelin enwrapping it, and this is also how it looks like in the normal appearing white matter. And one of the reasons that my lab is very interested in oligodendrocytes is because in diseases as multiple sclerosis, what you have is an autoimmune attack targeting the myelin, leading to demyelination, and then the transmission of the electrical impulses by the neural circuitry becomes much slower, and that ultimately leads to the neurological symptoms that the patients in MS have. Can you see my mouse?
Yes, I can see.
Yes, okay, great. So oligodendrocytes are originated during development around embryonic day 12.5 in the mouse from neural stem cells. And then these embryonic oligodendrocyte precursor cells, they proliferate during embryogenesis, and they only differentiate at postnatal stages. Then when the neuronal circuitry starts to be established, and then they start myelinating neuronal axons. Now, there is a population of these progenitor cells that throughout my presentation I'll call OPCs that remains in the adult brain and spinal cord in an almost quiescent state, dividing very slowly, but that can be activated to proliferate and differentiate and then producing myelin. And one of the occasions that this happens is in relapsing remitting MS. So in multiple sclerosis, many of the patients in the beginning, they have symptoms, but then they recover. And they have these cycles of recovery, symptoms and recovery. And one of the reasons of this recovery is that oligodendrocyte precursor cells can migrate to the site of lesions, differentiate and start producing new myelin, which leads then to the recovery of the transmission of the signal, of the speed of the transmission of the signal by the neural circuitry. However, many patients evolve to a phase of the disease, which is a progressive stage or a chronic stage, where there is no recovering of the symptoms. And this is despite that there might be oligodendrocyte precursor cells in the lesions, but they fail to differentiate and produce myelin. And one of the main questions in my lab has been why do these precursor cells fail to differentiate and produce myelin in the context of progressive MS, and whether we can induce them to remyelinate in the context of disease. For this, we have been investigating the transcriptomes of these different cell populations of oligodendrocytes during development and also in disease. And one of the first studies in my lab was performed by a postdoc in the lab, Sueli Marques. At that time, together in collaboration with a postdoc in Amit Zeitzel in Stan Linashon's lab, here also at Karolinska Institute. It's a lab next door to us, and Stan's lab is one of the pioneers of single-cell RNA sequencing. And what Sueli and Amit did was that they performed single-cell RNA sequencing in different regions of the juvenile and adult brain or in spinal cord, in dorsal and ventral regions, in a total of 10 regions, analyzing 5,000 oligodendrocytes. Basically what they did at the time is that they dissected these different regions, prepared the whole cell suspension, then used at the time the fluid MC1 microfluidics device that would allow to isolate every single cell in this compartment, and then perform single cell RNA sequencing with a technique that Stan's lab had developed, START-seq1. And I mean, if you are interested in the different technologies of single cell, I can go in more detail at the end of the talk at the questions, but now I'll focus more on the biology. So after doing the single cell RNA sequencing, Amit developed a clustering algorithm that allowed to put the cells in groups, in clusters that have similar transcriptional profiles. And you can visualize this in this heat map. In each column is a single cell, and each row is genes with different expressions. And you can see that all these different cells in these different clusters have this different expression of all these different genes. Another way to visualize this data is by showing this t-SNE representation of the data. So every data point in this data set is is multidimensional. So you have a single cell with different vectors of the expression of the different genes. So one way to represent the data in a 2D manner is to reduce the dimensionality of the data. And then here, every single dot corresponds to a cell, and cells that are close, that have similar transcriptional profiles, will be close together in these plots. So what you can see here is that we, for instance, have here a cluster that it's the oligodendrocyte progenitor cells. Then we have clusters of committed precursors, oligodendrocyte precursors, newly formed oligodendrocytes, myelin-forming oligodendrocytes. And then we identified 6 mature states of oligodendrocytes that were different at the transcriptional level. And this was one of the major interests in this study, because, as I've told you in the introduction, oligodendrocytes have been mainly thought to have one main function, which is to produce myelin. And this could suggest that oligodendrocytes might have additional functions that would be compatible with these transcriptional profiles. And this is something that we are very interested in our lab, and we have lines of research trying to investigate what could be the different functions of these different states of oligodendrocytes, or if they are only transitional states. But I won't focus so much on that in this presentation. I'll focus rather on our studies in the multiple sclerosis. And in particular, in our studies, again, performing single-cell RNA sequencing in a mouse model of multiple sclerosis, the EAE model. This work was spearheaded by Ana Falcão, a postdoc in the lab at the time. Now she has her own group, both in Portugal and also here at Karolinska, and with David Van Bruggen, a PhD student in the lab. And basically what Anna and David did is that they use tooled mouse lines where we would have GFP driven by PDGFR alpha, so then we could label the oligodendrocyte lineage. And then they induced this mouse model of MS, EAE, in these lines. And what you can see here in this graph is, so after the induction, around about 10 days after the induction, here you have the EAE score, the score of disability, and the mice start having the symptoms. And then here, what Anna and David did was collected the spinal cord at this time point, and then by fact sorting, isolate the oligodendrocyte lineage. And then applied a single-cell RNA sequencing using another technology called SMART-seq2. Again, I can discuss that more in detail afterwards, the technology. And here what you have is, again, a t-SNE representation of the data, where, again, every single cell is represented by a dot. And we identified, for instance, here, oligodendrocyte precursor cells, here, mature oligodendrocytes, also cells from pericyte lineage called vascular and leptomeningeal cells. And we actually have also some microglia, which we don't know if it's because PDGFR alpha is expressed transiently during the microglia lineage, or if it's because they were phagocytizing GFP particles. but anyhow, we got it in our fact sorting strategy. And as you'll see a little bit later, they were actually quite useful for our analysis. And then what David then did is again doing this clustering analysis to see if he could find groups of cells, clusters that have similar transcriptional profiles. And what we could indeed see is that like they were, he could identify these clusters, and they were, some of them were specific for EAE. Here in green you have control, cells coming from control, and in purple cells coming from EAE. And what we could see was that there was a clear distinction of the progenitors in both EAE and control, and also of the mature oligodendrocytes. Now, if we go look in more detail, what we could see is the expression of different markers characterizing these different populations. So here in the dotted line, you have the control populations, both mature oligodendrocytes and progenitors. And here in the solid line, you have the clusters coming from the EAE. And here you have several markers that are specifically present in progenitors in the mouse model of MS. And for instance, one of the markers is MERV, which is a transcription factor that is involved in differentiation, indicating that progenitor cells are initiating the process of differentiation in the mouse model of MS, which is expected from the literature. We also see, for instance, an increase in cycling OPCs with Ki67, again, suggesting the OPCs are being activated in the mouse model of MS. And then we also saw several markers specific for different mature oligodendrocyte populations in the mouse model of MS. For instance, one of the classes of genes that we saw was serpins, which are involved in survival of oligodendrocytes, and we saw that specific subpopulations had increased expression of serpins. And for instance, specifically this serpin A3N, we went to the tissue and validated and saw that indeed in control spinal cord, we didn't have by RNA scope in situ hybridization, we could not see much expression of serpin A3N in oligodendrocytes, which are SOX10 positive. But then in the mouse model of MS, we could see an increase on serpin3N, which then validated our single cell results. So we also were having this data set of cells of the oligodendrocyte lineage in the mouse model of MS, and also genes that are specifically expressed in these populations. We, at the same time that we were doing this study, it came a study from the International Multiple Sclerosis Genetic Consortium with a new list of susceptibility genes for MS. And this was uploaded in bioRxiv in 2017. And it was then published in 2019 in Science, the paper with this list of genes. But from the bioRxiv, what we could then do was compare this list of susceptibility genes that had been suggested by International Multiple Sclerosis Consortium and the genes that were expressed in our different populations. And what we could see, for instance, is that, for instance, in our microglia population, we could see the expression of some of these susceptibility genes as expected. And we could see, for instance, H2AA, that some of our mature oligodendrocytes in EAE also seem to express it. But what was the most interesting finding is actually the non-MHC locus associated susceptibility genes, where we saw that, again, they were expressed in microglia, which was one of the main conclusions of this paper. But actually, we saw that in mature oligodendrocytes, there were subpopulations that also express some of the susceptibility genes. But even more interesting was that actually our control progenitor population also expressed several of these susceptibility genes. And what we think this could imply is that usually susceptibility in MS is linked with the immune system, because many of the susceptibility genes in MS are immune genes. But what the expression of many of these susceptibility genes in OPCs could suggest that the susceptibility might not be reflecting in a dysfunction of the immune system only, but actually could also be reflected with a dysfunction on the oligodendrocyte lineage. And this is something that we are also quite interested in investigating. But what we also found in our data set was that, and like Jorge mentioned before, was that many of the both OPCs and mature oligodendrocytes, we saw an increased expression of major histocompatibility complex genes, class 1 and class 2. And why this was very interesting was because MHC class 1 and 2 are usually linked with antigen presentation, and specifically for MHC class 2 is usually expressed in antigen presenting cells like B cells or macrophages that then present and then activate T cells. But oligodendrocytes or OPCs had not been previously thought to be antigen presenting cells. And this could suggest that in the context of a mouse model of MS, you could have a transition to a state where they would express these genes. Now, one thing that is quite important to think about when you do single cell RNA sequencing is that you are sequencing the transcriptome of one cell, but in the dissociation process, there can occur some artifacts. Like you might have like doublets of two cells sticking to each other, and we could be that this profile that we are seeing could be a OPC or a oligodendrocyte together with an immune cell, and that's we would sequence them together, and then we would get the profile of both cells. And bioinformatically, we can take care of some of these doublets, but it's still a concern that we would have that in the single cell studies. So this is why it's very important in single cell studies to do validations. Again, what we did here, we did validations with RNAscope, in which, and what you can see here in red, you have CD74, MHC class 2 marker, and then IF1, a marker for microglia. And you see a lot of those cells double positive for both, but then you see also cells like this one, which is positive for in green, which is SOX10, so it's an oligodendrocyte. It also has CD74, but does not have any IF1, so it's not a microglial cell. When we did the quantification of these cells in lesions in the MS model, in the EE model of MS in the spinal cord, we found that around 4% of the oligodendrocytes have CD74 and have MHC class 2 markers, suggesting then that this is indeed not only an artifact of the single cell RNA sequencing, but that also we could see it in the tissue. Okay, so one thing that we were then quite interested is like, okay, so in the context of EAE in the mouse model of MS, OPCs and oligodendrocytes can transition to this immune-like state. How is it induced? And actually, so what Anna did was that she went back to the literature and she found some papers in the late 80s and beginning of the 90s where actually oligodendrocytes and astrocytes have been treated with interferon gamma and have been seen by immunohistochemistry to express MHC class 1 and in some cases MHC class 2. But this was very controversial. And after that, in tissue, people had not seen it again. And then it was disregarded and considered more that oligodendrocytes and OPCs could not express these kind of markers. So what Anna did was actually, and at that time, one of the papers that Anna saw was a paper where they've treated rat oligodendrocytes with dexamethasone and interferon gamma, and in that case, they could see the expression of MHC class 2. So what Anna did was that she repeated the experiment using SOX10 GFP mice, where she could sort the oligodendrocyte cells or OPCs, and then treat them then with dexamethasone and interferon gamma. And what she could see was indeed that she could see by both immunohistochemistry and RNAscope an induction of MHC class 2 genes upon treatment with these two compounds. What Anna also did then was did qPCR to try to distinguish what dexamethasone and interferon gamma were doing. And then what she could see was that actually the immune genes are mainly induced by interferon gamma, while other class of genes are induced by dexamethasone. Pointing out that interferon gamma seems really to be the major player to inducing an immune OPC or oligodendrocyte phenotype. Now, I mean, our lab is not a neuroimmunology lab. So then we started, when we had this finding, we started collaborating with André Cacais, here also at Karolinska Institute, who is in Thomas Olsen's group, and they have a lot of experience with neuroimmunology. And then Andrea, together with Anna, did a series of experiments to try to see what was the function of these immune OPCs. And one of the experiments that they did was to label myelin with pH rhodo, which is a dye that only fluoresces within acidic environments like the lysosome. And when Anna treated this pH rhodo myelin, gave it to OPCs, then she could see that OPCs would incorporate phagocytize the myelin, and then the myelin would start fluorescing, indicating that indeed OPCs are capable of phagocytizing myelin. And then in another experiment that Anna did together with Andre, what they did was they did co-cultures of OPCs, treated or not with interferon gamma, with T cells from the TD2 mice that have CD4 T cells that are specific for the MOG antigen. So they did these co-cultures of the OPCs, CD4 cells, and with the MOG antigen, and then assess by FACS sorting, FACS analysis, what was the behavior of the T cells. And what they could see is that specifically for the memory T cells, they could see an increase in survival and proliferation upon treatment with MOG, with or without interferon gamma, suggesting that OPCs seem to be able to present MOG to the T cells and then lead to their survival and proliferation. And in a sense, at least in vitro, they seem to be able to activate the T cells. So we published this study in 2018, and actually already in 2019, there were a few other groups that had similar findings that similar to ours. First, the group of Peter Calabresi in the US, and he's at the time PhD student Leslie Kirby, where they showed that indeed MHC class 1 and 2 can be induced in OPCs, and then in their system, they showed that it leads to CD8 activation instead of CD4 that we saw in our study, and that this would lead to a negative feedback loop that leads to the death of the OPCs. And another group, Alban Gauthier, also in the US, had a similar study where also they saw this. So it seems several groups are now seeing this aspect of OPCs that seem to have, like, at least in mouse, in the mouse model of MS and in mouse models of disease, that seem to be able to transition to a state of immune state that seems to be activating immune cells. We were also interested to see if this was happening in humans, and this is something that we postdoc in the lab, and Eritz Aguirre did in collaboration with Sarah Jekyll, a postdoc in Anna Williams and Charles French Constance Lab, University of Edinburgh, also in collaboration with Roche in Basel and Diraj Malhotra. And basically what Eritz and Sarah did was that they performed single nuclei RNA sequencing in post-mortem white matter samples from 5 individuals without neurological disease and 4 individuals with MS. So we found, one thing that we found is that we found heterogeneity of oligodendrocytes also in human, and that there seems some populations that seem to be saved and others more susceptible in MS versus control. But what we also find in this data set was a population which we call immuno-oligodendrocytes, oligodendroglia, which express many of the genes that we had seen in the mouse model of MS. And we also then, Sarah did a validation, and she could see also by immunohistochemistry also the expression of oligodendrocytes with olig-1 positive that also were positive from MHC class 2. Indicating that this immune oligodendroglia is not present not only in the mouse model of MS, but we also can see it in patients, in MS patients. So basically, these single cell studies allowed us to revisit a little bit the landscape that I've showed you in the introduction, and indicate that oligodendrocytes, apart from producing myelin and myelinating axons, also can transition to immune oligodendroglia state, which seems to be able to modulate immune cells. So one of the questions that we are also interested in my lab is how at the chromatin level these changes are of transcription are induced. And basically we are interested in to see how epigenetic information in the form of epigenetic marks like histone modifications, DNA methylation and so forth is deposited in the chromatin and allows expression of myelin, for instance, of immune genes specifically in the immune oligodendrocytes. But like in wild type OPCs, whether how the chromatin is different and then does not allow the recruitment of the RNA polymerase and transcription of these immune genes. And in order to do this, we have been using a technique called single cell attack-seq, which is a technique that allows to see how is the chromatin state in single cells. And basically, so if we go back to the previous slide, basically, in order for a gene to be transcribed, at its promoter or its regulatory regions, the chromatin has to be open in order for the RNA polymerase to be able to go in and then start transcribing that gene. So one way to see how the chromatin, so there is this technique called attack-seq where you can actually assess whether the chromatin is open, allowing the RNA polymerase to bind, or if it's closed and then preventing the RNA polymerase to bind and then preventing transcription. And this technique is based on the fact that this enzyme TN5 can access the open chromatin and then cleave the DNA and insert Illumina adapters, by which then we can use to sequence, and we only sequence the regions of the chromatin that are open. So what Mandy, a PhD student in the lab, and Eritz, a postdoc in the lab, they did was that apply this technique at a single cell level, again in the mouse model of MS, and focusing in oligodendrocytes at the peak of the disease. Using again another technology which is 10x provided by 10x Genomics. And again, so here, the cells that we have done the single cell attack-seq, then what we would try to do is do the clustering, see cells that have similar chromatin profiles, similar profiles of chromatin accessibility genome-wide. And again, here is a Disney representation of the data of the cells, as you've seen before. So we could identify several clusters of cells. And then when we try to see which ones come from control and which ones come from EAE, what you could see already here is a clear segregation of cells coming from control here, and cells coming from EAE here, and here. Now, what we want to know is what kind of cells, what is the identity of these cells. And in order to do this, what Eritz did was actually use the single cell RNA sequencing as our background, as our template, and then try to infer which were the populations in the single cell attack-seq dataset. So basically, in the single cell RNA sequencing, we know which are the population and which markers, which is the gene expression of these different clusters. So what Eritz did was she used label transfer, and so she picked the single cell attack-seq dataset, and she went to the promoters and saw, okay, regions around the promoter are open. So this suggests that these genes might have a gene activity. So she called a pseudo gene activity based on the attack-seq data. And with that, then she could compare the attack-seq data with the RNA sequencing data. And with that, she could see that, for instance, this cluster here had a pseudo gene expression profile similar to the mature oligodendrocytes, this one to the OPCs, and this one to the microglia. And she could go even into more detail and subcategorize the cells that we had from the attack-seq to the cells that we had seen with the identity that we had previously seen in the single cell RNA sequencing. And again, we could also see that using this label transfer approach, that again, we had mature oligodendrocytes that were specific for control on the AE, OPC is also specific for control on the AE, and the microglia was mainly in the mouse model of MS as expected. Now, if we plot this pseudogene activity from the ATAC-seq also in the TSNA, you can see here, this is a marker of PTPRZ1, which is expressed in OPCs, and we see that it has much more pseudogene activity in OPCs. This is MOG, which is expressed in mature oligodendrocytes, and you can see that it's mainly in mature oligodendrocytes, but not so much in OPCs and microglia. And then we have IFIT1, which is expressed in microglia, and we can see here the pseudogene expression in microglia. But what we could also see was that there seems to be open chromatin or pseudogene expression also in the mature oligodendrocyte, specifically in the mouse model of MS. So what we then went to see is that if we picked each one of these populations and then try to see where we were seeing this increase in the, which genes had increased accessibility in control versus EA. And both, either in OPCs, mature oligodendrocyte 1s and mature oligodendrocyte 5 and 6, what we could see is that these were mainly genes involved in immune responses. So, and again, for instance, one of these genes is this gene's MHC class 1 gene, PSMP8, which again, we see increased accessibility of gene or pseudogene activity in mature oligodendrocytes, but also in microglia. So if we look actually at the genome, you can actually see now this is the locus of PSMP8, here is the promoter, and this is how these are the reads that show that this region has higher accessibility. And what you can see here is, for instance, that mature oligodendrocytes in control don't have accessibility at the promoter of this gene, but that they have increased accessibility in the mouse model of MS. But what we also, so indicating that in the oligodendrocytes, not only we see this change in transcription, but actually the chromatin also changes and opens up to allow the transcription. But what was also quite interesting is, for instance, this other immune gene, TAP2, what we could see is that there is already accessibility in control OPCs and mature oligodendrocytes. And this here you can see it here also in the pseudogene expression activity from the ATAC-seq, where we can see that there is an open chromatin everywhere. So this suggests that not only there is a transition to open to chromatin of immune genes in the mouse model of MS, but actually that the chromatin in some of these genes or immune genes is already open in wild-type oligodendrocytes and the progenitors. So what we then did was that we picked genes that had increased expression or decreased expression in our single-cell RNA sequencing for control versus EAE, both in OPCs and, for instance, in these subpopulations of mature oligodendrocytes. And then we went to see how was their chromatin accessibility state in ATAC-seq, comparing again control versus EAE. And what we found was that there is several genes that have increased expression in the RNA sequencing that also have an increased accessibility of their promoters or regulatory regions, both into two populations. But what was more interesting is that we have these genes that have increased accessibility, increased expression, but the accessibility is the same, indicating that they are already in a prime state. So the OPCs and mature oligodendrocytes during development already have a chromatin that is ready to be activated to initiate transcription, and that has a chromatin open. Which is quite interesting. So it introduces this concept of primed immune genes in oligodendrocytes during development. We also were interested in analyzing the data in a different manner, which was usually, in order to be these regions of open chromatin, they have transcription factors that bind and then recruit the transcriptional machinery to lead to the transcription of the RNA. So one way to analyze the data is actually to see differential accessibility of transcription factor motifs. And by doing this analysis, we actually could see here in each row, each column we have a cell, and each row we have different transcription factors. And what we can see is that the accessibility of SOX transcription factors is enriched in cells of the oligodendrocyte lineage, which is consistent with what would be expected from the literature. And by doing this kind of analysis, we actually could use like the transcription factors accessibility to try to infer, to do clustering of the data, and also try to see whether we could, to see if we could separate in different clusters. And indeed, we could separate clusters coming from OPCs, mature oligodendrocytes, or microglia. And again, we could separate them from EAE versus control. Now, if we look now within these clusters, what we can see is, for instance, that here is a transcription factor that is usually present, acting in microglia, and we can see that it has higher motif accessibility in microglia. But then we also have this transcription factor like BAC2, that it's also an immune transcription factor, but that we also see increased accessibility in this population. And this population is a population of mature oligodendrocytes. And this is the kind of transcription factors that we are quite interesting, which might be transcription factors that might have a function to regulate the transcription of immune genes in oligodendrocytes. And we want to see whether they indeed have a function to activate these immune genes. Another interesting thing that when we do this analysis of transcription factor motif accessibility is actually that of these transcription factors that we see increased accessibility both in OPCs and mature oligodendrocytes, some of them have increased expression, but there are others, like for instance here SOX8, that the expression between control and EAE is similar in mature oligodendrocytes, which would then suggest that by expression, we would not suspect that it would have a role in the regulation of these cell states. But actually, when we look at the transcription factor motif accessibility, we see an increase, suggesting that even if the expression level of this transcription factor, SOX8, in control oligodendrocytes and EAE is similar, actually it might be more active in EAE, regulating the immune genes. Okay, so now for the last part of the talk, I'm going to focus on, again, on interferon gamma. And as I've mentioned you before, interferon gamma, we've seen in our previous study that interferon gamma is inducing the expression of several immune genes in oligodendrocyte precursors and in mature oligodendrocytes. So we wanted to see how interferon gamma is inducing changes in the chromatin in immune genes in oligodendrocyte precursors and mature oligodendrocytes. So one of the first things that Mandy and Aneritz did for this was perform RNA sequencing, bulk RNA sequencing in OPCs treated with interferon gamma, and also did a TAC-seq. And what they could see is an increase of several immune genes upon treatment with interferon gamma, as we had seen before by qPCR and RNA-scope. But what was interesting is actually at the chromatin level, interferon gamma does not seem to, it induces the increase of accessibility of certain genes, but not as extensive as the RNA sequencing. If we visualize this in the genome browser, this is like, again, the MHC class 2 gene, where here you can see that by RNA sequencing, the increase in expression upon interferon gamma treatment, here, both here and here. And then in the TAC-seq, we see in this gene that there is an increase in the accessibility, the chromatin accessibility, also in these genes. But then we have other class of genes where, again, with interferon gamma, by bulk RNA sequencing, we see an increase in expression. But then by a TAC-seq, what we can see is that some of these genes do not seem to have a dramatic increase of accessibility. And this is, for instance, an example of MHC class 2 gene, where in the single cell TAC-seq data, we had seen that there was increased, that the accessibility was not changed. And here with interferon gamma, we also see that the accessibility is not that much changed, but it's changed actually in other loci. So this could suggest to us that perhaps what we have observed in the single cell ataxic data that was also could also apply to interferon gamma treatment. So then we also did the same comparison. We had genes that were increased accessibility with interferon, increased expression with interferon gamma, and we in some of those we see increased chromatin accessibility, but we also saw many genes that had the same state of the chromatin. Again, suggesting that the chromatin state is already poised or prime in wild type OPCs to, and that just needs another stimuli to induce the expression of these immune genes. So far what I've talked with you is just the opening of the chromatin, whether the chromatin is open, the nucleosomes are separated in the transcriptional start site to allow transcription. But there is several other layers of regulation of this process. And like I mentioned in my introduction, one of them is histone modifications, that post-translation modifications at the histones that also can regulate if the chromatin is more open or closed, and that also can recruit the transcriptional machinery to allow transcription. Years from the last decade has shown that like there is specific modifications that are associated with transcriptional activation, like H3K24/3 methylation at promoters, and others like H3K27/3 methylation that is usually involved in repression of transcription. And then there are others like H3K27 acetylation that are present in enhancers, and that usually is associated with active enhancers. So what we've decided to do is again try to see what kind of changes interferon gamma induces in these different modifications, also in transcription factors binding. And again, here you have an MHC class 1 and an MHC class 2 locus. And then what we, for instance, here we use a technique called cut and run, which is allows you to identify genome-wide the presence of some of these marks. And I can again talk more in detail about this technique. And what we could see is that, for instance, here we have control and here interferon gamma, is that like, for instance, this mark for enhancers is increased in this MHC class 1 and 2 loci upon interferon gamma treatment, indicating that there might be some more active enhancers in this region. And what we also profiled was CTCF, which is a transcription factor that is involved in chromatin architecture. And again, what we also could see is that there is more binding of CTCF in this region, suggesting also that there is a remodeling of the chromatin architecture in this region. And using the ataxic data and H3K27 acetylation, we could actually model the interactions between promoters and enhancers, the regulatory regions. And we could also see that there is an increased interactions between a remodeling of the chromatin architecture and more interactions between enhancers and promoters upon interferon gamma treatment. As I told you before, there is also these other marks, H3K4/3 methylation and K27/3 methylation, that are usually associated with activation, the K4, and repression, the K27. So again, we went to see how were their profiles upon interferon gamma treatment. And one thing that was striking from the beginning is that both in the MHC class 1 and MHC class 2 genes, the repressive mark was already present in normal wild-type OPCs, but the activating mark was also present. And this is like suggesting that, and this is the activating and the repressive mark suggests that the cell have both, the OPCs have both information to activate and repress, and this is called as a poised prime state. And but that we can see upon interferon gamma treatment is that the repressive mark is downregulated and the activating mark is upregulated. So it seems that interferon gamma really leads to a change in the chromatin, these chromatin marks, in order to allow to the transcription. And again, if we look like genome-wide, we do see that interferon gamma leads to the reduction of several of the H3K27 mark in many genes genome-wide, while it induces the increase of H3K43 methylation in many other genes. So the hypothesis from here is that interferon gamma seems to be modulating the deposition, or actually the removal of this repressive mark in these immune genes in OPCs. So what we then, Mandy and then Eritz did was that they treated primary OPCs with an inhibitor of the enzyme that deposits the mark, EZH2, and then treated with interferon gamma. So here you can see upon treatment with the inhibitor, you can see a reduction of the mark. And then when we treat with the inhibitor and then we do RNA sequencing, we can actually see that there is an increase in the expression of some of these immune genes, a little bit in control conditions, but then upon interferon gamma, there is really a synergy in some of these genes, indicating that so preventing the deposition of this mark, H3K27, synergizes in interferon gamma for the activation of these immune genes. And that when we did gene ontology analysis, we did indeed see that this was induced, that most of these genes were like involved in immune processes. So then what Mandy and Eritz did was actually the opposite experiment. So the hypothesis that interferon gamma would be leading to the repression of to the removal of the H3K27 methylation mark. So then they use this. Okay, my mouse just died, so I'll just try to. This mark H3, this Jumonji-D3 is an enzyme that removes the mark, the H3K27. So they use an inhibitor of this enzyme, which then allows more of the mark to be present, so prevents the removal of the H3K27 repressive mark. And what they could see was that upon interferon gamma treatment and addition of this inhibitor of the removal, they could see less activation of the immune genes, suggesting then that the removal of the mark is really one of the processes by which interferon gamma is letting the OPCs be in a poised state and preventing to express immune genes. Okay, so now to finalize, I hope that within my presentation I was able to convey the message that oligodendrocytes, that single cell omics technologies has allowed us to unveil new aspects of oligodendrocyte biology, both in development and also in multiple sclerosis. Specifically, as you can see here in this scheme, you have like a OPC that if you go to the left, it goes to the normal process of development and myelination, but that we show that in the context of MS, it can by probably the presence of several immune cells that are induce a change in its transcriptional profile into an immune profile. And then this immune OPC on the oligodendrocyte can then modulate other immune cells. And also that like our epigenomic data, single cell epigenomic data seems to suggest that oligodendrocyte precursor cells and oligodendrocytes already are primed and are poised to go to this immune state. And that modulation of chromatin enzymes like polycomb is very important to put the brake to prevent them in the normal scenario to go to this immune state, but that in the context of disease, factors like interferon gamma lead to the transition to an immune state. Okay, and with that, I would like to finish by thanking my lab. Here is the current lab. I mentioned most of the people that were involved in the work, mainly Eritz and Mandy now for the epigenomics, together with Mukund that did a lot of the epigenomics works also. And then also Anna Falcao and Sueli that did many of the work previously. We have collaborators that were involved in some of this work, Stanley Nashon, Charles French and Anna Williams in Edinburgh. Dheeraj Malhotra in Basel, and also Howard Chang in Stanford that helped us in the beginning with the single cell attack-seq, and also all our funding agencies. Okay, and with that, I'm finished, and I'm be very happy to answer any questions that you might have.
Okay. Now I will activate. Let's see, activate everyone. So please turn off the microphone. Those that don't want to ask anything, and just
Trying to see how I
go ahead with questions.
I'll stop sharing.
Lorena, how are you?
Hi, I will turn on my camera, maybe. Hi, Gonzalo. This is a very interesting talk. I wonder how general is this process of becoming an immune antigen presenting cell? Do you think that inflammation promotes this change in phenotype for cells, local cells in any tissue, or is it something that's specific for glia or specific for oligodendrocytes? Is it a general process or is it oligodendrocyte specific?
Well, in our data set, we can, we also have in our data set these VLMCs, which we think are from the parasite lineage. And we also see the expression of MHC class 1 and 2 there. What we don't know is if interferon is inducing them there. In our data sets, we don't have neurons, so we don't know if neurons, for instance, would be able to do that. I mean, it's known that Schwann cells have the capability to express MHC class 2. So at least some glial populations and astrocytes also.
Right.
So I think some I don't know if we can generalize to a lot of populations. I mean, there was also at the same time that our study came, there was also a few studies, for instance, with intestinal stem cells indicating the same. So I think in the body there is several inflammation might trigger some populations to start expressing these immune genes and then having a separate immune function. Now, the other question was whether this is specific. I think you were asking also if this is specific for MS or?
Sorry, I missed the last part because my
I think you were also asking if it was specific for MS or no. Okay, I'll continue in those lines.
No, I was asking whether, yeah, okay.
So, Lorena, yeah, you can
Just send the chat. You can send the chat, and then you can
yes,
reply.
I think can you hear me now?
Yes.
I think I reconnected.
Yes.
No, the question was whether any inflammation environment could trigger more like a general process. Maybe MS is one particular case of this, but no.
Yeah. So I mean, we actually in our human study, we actually see it also in our control individuals. We also see the oligodendroglia. And actually, when we looking at other data sets of single cell RNA sequencing data with aging, we also see them there. So what we think is this is a general process that with aging and inflammation can occur. So I don't think it's specific for, and then also in Alzheimer's disease, in some of the data sets with Alzheimer, we also see the expression of some of these genes. So I do think that this is more a general process, but then in the different contexts, it might be activated in a different way.
I see. And one more, maybe I missed in your graphs, how different are these immune cells from an immature, earlier, more immature precursor in the lineage? Is it something different than immature or OPCs? How different is that? Because I saw that some of the genes that you showed that changed in accessibility, for instance, or expression level, followed the pattern that more immature cells showed. Maybe.
So I would say, I mean, what we are seeing is that it's basically, they have all the markers of OPCs, for instance, but then they just turn on some specific gene programs for immune cells. And then
but
the
some
same
of those
thing.
immune genes were also present in the, some of those immune genes were also expressed, if I
So the immune genes were not expressed in the OPCs, but
see, okay.
but what they have is that the chromatin is in
Right.
some of them is already open. So
Right,
basically, we
okay.
are just waiting for the stimuli for them to be activated.
Okay.
Okay.
So Jorge Correales has a question.
Hi. Very nice talk. Thank you very much. My first question is if you, in addition to the MHA1 and class 2, did you find the OPC co-stimulatory molecules that support the possibility that this oligodendrocyte can present antigen in a productive way?
Yes, well, we indeed did, and actually that's a quite interesting question because we do find both co-stimulatory and co-inhibitory molecules. So now the question, and we are investigating that in the lab, is depending on the context, it actually might have one work to activate immune cells or actually to prevent immune cell activation. And at least in vitro, we are seeing that it's activating immune cells. But, and this was with CD4 T cells that we have studied. But now we are also expanding to other kind of immune cells, and also we are also doing studies in vivo to try to see what could be the function in vivo.
Okay, thank you. And the other question is, when you consider that your oligodendrocyte, precursor oligodendrocyte, have not a chance to remyelinate progressive MS cases, do you take in account that there is also a factor for the axons? So in the progressive MS, the neurodegeneration is an important factor that for most people is the main condition for the progression, more than the remyelination. So do you take in account this point?
No, I mean, so yeah, it's a very important point, and it's I mean, in our studies we usually do not focus. I mean, we haven't focused so much in the neurodegeneration, but of course it's a very important aspect, because if there is no neurons to remyelinate, there won't be any point of having the OPCs. I think the point that I was trying to make more is that, and this is actually not data from us, but more from Peter Calabrese's lab, is that what they've seen is that once the OPCs transition to immune OPC state, they do not, they differentiate less, suggesting that it in the path for remyelination, this transition to immune state might contribute to less differentiation and remyelination. But now there is other questions that it's also, I mean, like you were mentioning, neurodegeneration. Can it be that if these immune OPCs are presenting antigens, are they presenting also cross-presentation of antigens from axons and so forth? But that, again, we don't know.
Okay, last question. This heterogeneity that you found in the OPC, do you think that it's also different, they have also different functions?
I mean, in the OPC, we actually do not find much heterogeneity apart from this immune phenotype. We found it more in the mature, but again, it's we are still investigating. I mean, for instance, we have now a study that we see that there are some populations of mature oligodendrocytes that we only see in the spinal cord and very few in the brain. And we also, that seems to be some preference for different kinds of ascending and descending tracks in the spinal cord. So we think that the function might be related with the kind of neurons that they are interacting with. But we are not sure. We also are seeing that, for instance, this population seems to be more susceptible to spinal cord injury. But for instance, for EAE, we don't see susceptibility for this population. So we think that there is some functional difference, but we are still investigating what it could be.
Thank you very much.
Right, then we have a question from Juan. Yeah, so Yes.
Yo tengo una pregunta.
Yes.
Hi, Gonzalo.
Yes. First,
So-
Juan, and then you, Marcelo.
Okay.
That was a very nice talk.
Thank you.
So I have two questions. So the first one is, did you check if using your transcriptome analysis, if you have new mutations in these progenitor cells that could be working as neoantigens and starting like an immune response that increase the secretion of interferon gamma, and that is like, how is the story starting? That's my first question. Where is coming that interferon gamma? And
Well, the interferon gamma, we don't well, we know it comes from immune cells. We think it's T cells, but we are actually investigating several sources that it comes. And one of the reasons we think is because there was also a recent paper from Anne Brunet last year where they showed that in the aging mouse, T cells can go through the blood-brain barrier and then go to neurogenic sites, release interferon gamma, and induce also kind of an immune phenotype in neurostem cells. But we don't know exactly how it would happen in the case of disease. I mean, so there is several scenarios here. Either that the OPCs, there is an inflammatory event that turns OPCs into immune OPCs, and that triggers a cascade that eventually would lead to the activation of the peripheral immune system, or that the immune OPCs are actually just reactivating the peripheral immune system response. But this is something that we are investigating in the lab. So it's at this point, it's just hypothesis that we have.
okay, that's nice. The other question is, when you have a promoter in a poisoned state, usually you have both enzymes, the methyltransferase and the demethylase York-Shumonshi and the ECH2 of the polycomb repressor complex in a balance. And usually for some signal, that balance is broken and you turn in one direction and in the other. So you induce a gene or you shut up a gene. So the
Yes.
question, did you make any study about which is the mechanism I don't know if there is a transcription factor or some kind of phosphorylation or another modification that is shutting down the ECH2 activity and turn down the Shumonshi D3 activity?
At this point, we don't know. I mean, but it is something that we are quite interested to see. I mean, I think some of the transcription factors that we are now focusing on, it would be interesting to see if they are interacting with Shumonshi D3 and if they are the ones that are recruiting it to the specific promoters and then leading to the demethylation. But this is work, this is work that we want to do in the future to try to connect one thing with the other.
Yes. Thank you.
Great. So Marcelo, sorry to interrupt, but Juan was lifting the hand like 10, 15 minutes ago. So
No problem.
Go
No
ahead.
problem. Thank you, Gonzalo, for your nice talk.
You?
So I'm not a player in this field, so probably I'm doing a not appropriate question. However, going to your very first slides where you characterize the OPCs expressing the MHC2 class 2. So can you discard completely that these MHC expressing cells are macrophages from the peripheral? My question is because when any presenting cells or a macrophage is invading a tissue, is eating and phagocytizing every tissue which is damaged in the surrounding. So if I remember well, you say that it's an OPC because it's expressing AFI. I don't know what is AFI.
AFI is a marker for microglia or macrophages.
Okay.
And I mean, so this is what, I mean, that's why we did the RNA scope, because I mean, I think the first thing that we thought when we saw the results is that it would be an artifact, exactly for the reason that you were mentioning. So that's why it was very important to go to the tissue and do RNA scope and then really see, and we do see some cases exactly what you say, that we can see a macrophage or microglia eating an OPC, very close to an OPC in eating. But these 4% that I mentioned are cells that we clearly don't see any nuclei around with any marker of macrophages or microglia, and that are isolated and that express the immune genes and the MHC class 1 and 2 genes and the oligodendrocyte markers. And then also there is a fact, I mean, transcriptionally, we would see it also in the single cell RNA sequencing data if they would have the profile of microglia or macrophages, which they don't. So, so we can differentiate that. But of course, that is, I think a subset of cells will be like that. What I think, so both the RNA-scope and now this data, the chromatin data really indicates that the OPCs do have the capability, and it's not all the immune genes, it's only a subset of the immune genes that they have the capability to activate and produce.
Okay, so perhaps it's important, I think one of our colleagues that before asked you about the importance to investigate more other co-stimulating factors in these cells. Thank you very much, Gonzalo.
Okay. So, Gonzalo, I have a question regarding these OPCs, immune OPCs, and how important are for the CD8 activation. I mean, it's known that the CD8 cells are the one playing a role in multiple sclerosis. So, do you think that it has an important role for this activation, or other kind of functions?
I mean, we don't know, but I mean, the thing is that the percentage of cells that we see, it's quite low compared to other antigen presenting cells at the peak of the disease. So that's something that we are trying to investigate now, is when do these cells arise, and whether it might actually be important in other stages, in the initiation, maintenance, and so forth. And at this point, it's all hypothesis. I mean, I don't think at the peak of the disease that they will be so important for the CD8, because I think there is so many other things, other cells that can that function. But I think like perhaps in the beginning of the disease, they might have a more important role. But again, it's only hypothesis. We need to test if this is the case or not.
Yes, because it's, I mean, is it not clear at all which is triggering the injury? So it's something from within the central nervous system or from the periphery?
Yeah, and I mean, that's a very interesting question. I mean, there might be a trigger that triggers oligodendrocyte cell death and then activates the OPCs, and then it starts the trigger. It can be, again, virus. I mean, there has been, so we don't know. So, yeah, we don't know.
Any other question? So comments? No? Yes, Juana?
Yes, Gonzalo, first of all, curiosity. When you are talking of adult OPC, are you talking about NG2 or general OPC?
I talk about PDGFR alpha. NG2, I'm a little bit ambivalent about NG2 because we do see NG2 and CS PG4 in the parasite lineage. And I mean, in our single cell data, that's very clear. So they are NG2 positive and PDGFR alpha positive. But then we have this other population, which is VLMC, that is also PDGFR alpha positive and NG2 positive, but which we see that it's from another lineage, which we think it's parasites.
And the other question is related to the Jorge question. Have you tested in another demyelinated models such as cuprison the presence of this special kind of OPC?
And so we haven't, but Peter Calabrese's paper, they did in the cuprison, and that's where they saw. So yeah, so they are also there, but yeah, so it will be interesting also to see if they are in other models.
The question is because cuprison model is not an inflammatory model, so there
yeah,
is
but
a big...
actually in the cuprison, it's the model that they have. It's cuprison, but then they also inject interferon gamma. Well, it's a mouse model of where they have interferon gamma expressed in astrocytes, and then they induce cuprizone, and then they see it there. What we wanted to see is actually to use Peter Stuy's model, where they have now the CAE model, which is a combination with cuprizone and EAE. But that's something that we want to test, whether we can see them there. But also what we are very interesting to see is more like, for instance, in Brian Popko's model, where they actually kill oligodendrocytes, and then to see whether that is induced in that system also. So, I mean, I think that in, I think in some systems we might see it, some systems we might not see it.
Thank you.
I have another question. So you show in some papers, some of the articles, that the subpopulations of parasites, also endothelial cells, are expanded in your model. So where do these cells come from? So where these endothelial cells or parasites? Do you think that they are CNS cells, or if they arrive from the periphery?
I mean, we think they give rise to parasites. When we had that paper a few years ago when we did lineage tracing, and that's how we saw that they were giving us the parasites. But actually, during the review process, one of the reviewers was asking that question, and the hypothesis that we generated at the time was that they would probably become cells coming from outside the CNS, migrating through the vessels, and then populating the CNS. But now there has been a few papers suggesting that if we differentiate human IPS or ES cells into the dopaminergic lineage, you get this population of ELMCs, which suggests that it's somehow in the neural lineage. And my suspicion now is that they might actually come from the neural crest. But again, so this would be my hypothesis here, that they are neural crest derived.
Okay, actually the parasites in the CNS, at least in the brain, they are neural crest. This is not.
Yes, so that's, I mean, they probably express very early on SOX10, and that's why we get them in some of our lines, transgenic lines, so.
Yes, but until now, nobody has shown that the neural crest can produce endothelial cells.
Well, they're not endothelial per se. They are
okay.
Parasites is
Okay, perfect. Okay, no more questions. Seems no more hands lifted or somebody. No, okay. So Gonzalo, thanks a lot. It was incredible talk.
Yeah, thank you very much for the opportunity and also for all the questions. Okay.
See you.
So have a nice day, all of you.
Thank you.
Bye.
Thank you, Gonzalo. Have a nice day, you too.
Bye.